Hello and welcome to Chapter 4, Part 1 for EFB103. This chapter is a tour of the cell. So here's where we start to look at cells, what they're made of, some of the basics of how they work. And the main topics here are listed below, and let's get started. So the first thing to focus on is this idea of cell theory. And this is basically states that the fundamental unit of life is the cell. All organisms are made out of cells. Some are just single cells, some are multicellular. The cell is the basic unit of structure and organization in living things. And there is a continuum or continuous line of descent from the first living cells. And this relates to this quote by Rudolf Virchow, omnis cellula e cellula, which basically means all cells from cells. So when it comes to studying cells, there are a few tools and techniques that we can use. One being a microscope, because when you're dealing with cells, most of these are not visible to the unaided eye. So in this little chart here on the right, we can see some examples of cells, say like a chicken egg, very big cell. You can see that with your eye. But when you get down to many other smaller cells that are typically found in your body or single-celled organisms in a pond, you need a microscope to see them. Now, one thing I highly recommend you check out are these two links on the bottom left that I have. This allows you to zoom in and out at different levels of magnification. The Learn Genetics one is sort of more focused on living things, molecules in living things and higher up. And then the NASA one goes down to like subatomic structures all the way up to the scale of the universe. Really cool web tools. But back to microscopy. Let's start with light microscopy. So this is probably what you've already done before in one of your classes. And you'll be doing that this semester in the lab that is a co-requisite for this class, using the microscope at some point. And so light microscopy is where you have a light source that passes through a specimen, and then the viewer is sort of on the other side of that light beam. The other two microscopy methods you see here are transmission electron microscopy. This looks similar to the light microscopy. Instead of using a light source, using an electron source, this allows finer resolution. Scanning electron microscopy also uses an electron source, but here you're actually looking at the surface of objects, and I'll show you some examples of that. But back to light microscopy. Or really with any kind of microscopy, there are three important parameters. One is magnification. That's sort of an obvious one, right? How much can you zoom in? The second one is resolution. These are all important, but resolution might be the difference between having a really good microscope and a not very good microscope. The resolution is that measure of clarity, how well you can resolve an object. So, for example here, I cut and pasted an image from a web page selling this microscope, this AMscope, and its magnification is touted to be 120x or 1200x, and this microscope's $45. That seems like a great deal for that amount of magnification. So that's all fine and good, but guess what? The resolution on this microscope is probably pretty low. So the magnification sounds good, but the resolution not so much. Take an example of, say, the research microscope in my laboratory. One of the lenses, the higher magnification lenses, might cost $800 on its own. That's not even the body of the microscope. So what you're paying for when you buy a better microscope is that resolution and the better lenses that come with that. Now, that doesn't mean that this cheap microscope isn't useful and maybe get some people engaged in science. You can definitely see some things, but if you really want to see stuff well, you're going to have to spend more than the $45. The third thing that helps us or influences what we can see with the microscope is contrast. So here are some examples. These are straight out of your textbook. You can refer to that as well. Look at bright field. This is the first box here. An unstained specimen. You can kind of see that there are cells there. Here's nucleus in the middle. But when we say stain the specimen, now that creates more contrast, right? We can clearly see a nucleus and these cells here. And we can use some other microscopic techniques, phase contrast, that basically allows us to get some shadow on the specimen. So now we see more. DIC is similar. We could stain with a fluorescent dye, for example. And we can use something even called confocal microscopy. I'll come back to that one. But these different methods allow us to have more contrast. All right, so keeping in mind resolution, magnification, contrast, how good can you get, or how much magnification can you get with a typical light microscope? Probably to about 1,000 times magnification. That would be with a 100x objective lens and a 10x eyepiece lens. And remember that cheap scope that you can buy online for $45, they're saying it can go up to 1,200x magnification. And that's really almost outside the range of what's possible. And in that case, you're just losing more and more resolution. Now, as we saw already, contrast can help. So you can use all kinds of staining techniques. Here is a nice example of a cell stained with a couple of different fluorescent stains. The red are sort of connected mitochondria with stained with this mitotracker red dye. Peroxisomes, we'll talk about those soon enough. Those are the little green dots everywhere. That big blue dot in the middle, that's the nucleus that's stained with something called DAPI. So there's all kinds of things we can do to help us in our efforts to observe and study cells. Let's circle back to that electron microscopy example. We want to look at smaller things or subcellular structures, things that are within a cell. We can use electron microscopy because our magnification and resolution is better at those really low levels. So scanning, we see a scan of erythrocytes, red blood cells here. Notice you're seeing the surface of them. So these things are fixed in place on, say, a piece of glass or something like that, and then coated with gold or some other substance, and then that's what you're seeing. Transmission electron microscopy, you have to take a very thin slice of whatever it is you want to look at. And then from there, it's almost like a light microscope. You're passing it through that electron beam through that thin slice, and then you can see these subcellular structures. Here's a nucleus of a cell. Here's the cytoplasm. There's little mitochondria everywhere. And so that allows us to see these really small things. All right, so what's an advantage of light microscopy over electron microscopy? EM sounds really good because we can see really tiny stuff, but what's an advantage of light microscopy? Given the choices you had, an advantage of light microscopy is the ability to view live specimens. With electron microscopy, you need to preserve and process the specimen before you can look at it. So you cannot view things live in real time. Other things you might have thought of are that electron microscope is very expensive. It's a big piece of equipment that takes up a lot of space, whereas, say, for a Gen Bio lab, a light microscope or many light microscopes are certainly feasible to pass out and everybody to use. The specimen processing is much easier with light microscopy, depending on what kind of staining you might be doing. But with electron microscopy, you need special equipment even to fix and process those samples before you can even look at them. All right, I said I would circle back to this confocal microscopy. This is a newer-ish technique. Here's a confocal microscope, pretty fancy looking. It's basically like a light microscope, but it uses computer processing to almost take digital sections or slices of the image. And if you see the example on the bottom right here, in panel A, what you're seeing would be a typical fluorescent image. It, looks pretty good, but you can see it's kind of blurry because if you focus on one part of the cell, Another part of the cell that's a little further down is just going to be a little bit out of focus, right? But what this does is it takes all those focal planes and then assembles an image, and then you get something like you see in B, where it looks just crystal clear at all those planes of focus. Another thing that we can do to study cells, especially the subcellular structures, is a technique called cell fractionation. And so what it does is breaks apart the cell. So you take a tissue culture here, blend it up. They show an actual blender here. There are things that look like that in the lab, but maybe use some other technique. And then you have this mishmash of subcellular structures and membranes, and then you throw them on a centrifuge, and this will spin at a certain amount times gravity, depending on how fast it spins. And that's what's going to happen is the stuff that's bigger is going to get pushed or pulled to the bottom of that tube. And then the little stuff is up in the liquid here. And so you have in the first spin, you have a pellet that's rich in cellular nuclei, so big stuff. So you take what's in the liquid portion here and you centrifuge it again, this time at a much higher speed. This is 20,000 times gravity for 20 minutes. Now we're pelleting, down the bottom here, chloroplasts and mitochondria. All right, let's take the liquid again. We'll save these guys and take the liquid and pass it to another tube. Now we spin it even faster, 80,000 times gravity. Now we're getting other, things that are smaller than mitochondria and chloroplasts. Those things are pelleting. And then we can do this again, pull off the liquid, save this, and then pellet the teeny tiny stuff like ribosomes. So this is a great way to separate out those bits of cells. So let's talk about the different kinds of cells. We can divide cells in a variety of ways, but we can split them into two main groups, the prokaryotes and the eukaryotes. So the prokaryotes are actually assembled of two different phylogenetic lineages, the bacteria and the archaea. And you can think of these are very much bacteria-like, whether they're archaea or bacteria. There are differences between them, but that's the idea. They're prokaryotes. Eukaryotes are things like you and I. One distinguishing feature of them is that they have a nucleus. So prokaryote basically means before nucleus. Eukaryote means true nucleus. Whether we're talking about a prokaryote or eukaryote, there are some common features that all living things have, that all cells have. So the usual top four would be, well, a cell membrane. Some of these things sort of sound obvious, but they're there. Cell membrane, cytoplasm or cytosol, DNA is this molecule of heredity, and ribosomes, these protein building structures. So those are kind of the four go-to common features of cells. But there are other common features. See if you can figure out which ones are common features of all living things with this question. Other similarities might just be the relative proportion of different chemicals in living things. So we're mostly made of water, and then approximately these proportions of other macromolecules. Let's look at prokaryotes first, and then we'll talk about eukaryotes. Obviously, they have all those unifying features that we've talked about. Some other things to note would be they have something called a nucleoid. So this is where the genetic information is. It's not a nucleus, but it's an aggregation of the DNA that's there. So it's not membrane bound like we see with a true nucleus, but it's a cluster of that genetic material. Prokaryotes have a cell wall. So there's a cell membrane, and then around that is a cell wall. Now it's worth mentioning the plasma membrane or cell membrane here, that this is kind of a gatekeeper for the cell. This keeps things in and keeps stuff out, decides what, quote unquote, decides what comes in and out of the cell. And this is a double layer membrane of phospholipids with proteins embedded in it. Now, relative to that plasma membrane is this concept of scale and surface area to volume ratio. So this kind of comes to the question of like, how big can a cell get, for example? You have lots of really small cells. So let's take the example of this little square here. The volume is 1, that's just our unit. We're saying 1 cubic units. And the surface area is 6. So the ratio, surface to volume ratio, is 6. As you get bigger, so you start to think of this maybe like prokaryote versus eukaryote, for example. Here's the total surface area, here's the total volume. Now the ratio is much smaller. And so this surface to volume ratio is really important for materials getting in and out of the cell. The bigger you get, the less surface you have for that to happen on. So being small is, there's some advantages to that. Things that are bigger maybe resist temperature and environmental changes a little bit better, just for example. So where you have the small cells, that surface to volume ratio is big. Larger cells, it becomes smaller. And as a consequence, what we see in eukaryotes is a lot of compartmentalization for processes to occur and to move materials around within the cells. We'll go over all these different compartments. These are called organelles. So let's throw a couple out here. Mitochondria, this is a rough endoplasmic reticulum. Here's a nucleus in the middle. Here's a Golgi apparatus. You can see these fine lines. These are cytoskeletal structures here in this animal cell. In a plant cell, here you've got a cell wall that's around it. Here, then the cell membrane inside of that. Here are chloroplasts. There's the nucleus again, and they have this large central vacuole in the middle. So if we can do some quick comparisons here, but let me ask you this. You've had a brief glance at these. What don't plant cells have? All right, so plant cells don't have that nucleoid. That was seen only in prokaryotes. And I've put together this table summarizing some of the features that these different groups of organisms have or don't have. And you can come up with something like this yourself. This can be a handy study tool when you think about what's going on in eukaryotes versus prokaryotes, and then plants versus animal cells. So the first organelle we'll talk about is the nucleus. Here it is pictured in this cartoon of a cell. If we look closer, the nucleus is a membrane-bound organelle. What's kind of interesting about that is it has 2 phospholipid bilayers. So the bilayer is, here's the 2 rows of phospholipids, and then there's 2 of them. So this is a double membrane-bound organelle. How do things get in and out of the nucleus? Well, they have these nuclear pores. This allows material to come in and out of the nucleus. In the center, or maybe not always, the center of the nucleus is this more dense area called the nucleolus. I'll come back to that in a moment. If we use a freeze fractioning technique of the surface of a nucleus, we can see the 2 lipid bilayers. So what happens here, you actually freeze cells and then kind of crack that frozen block of cells, and some of them are going to peel away this top layer of the nuclear envelope. So you can see here we're seeing the lower layer, the inner layer, here and here and here. That's the outer layer. And then these little dot, 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 dot everywhere, those are the nuclear pores. So the nucleus is that information central in a cell. That's where the DNA is found. DNA is organized into chromosomes. The DNA is wrapped around proteins. That complex of those 2 molecules is called chromatin. When a cell divides, that stuff has to condense. We'll see all this stuff as we go through the course. And what about that nucleolus? Remember that kind of dense part of the nucleus, the darker staining part of the nucleus? Why does it look more dense? It's because there's a lot of gene expression happening there. Gene expression producing a kind of RNA called ribosomal RNA. And this happens a lot. A lot of this stuff needs to be made on a regular basis because this is a fundamental component of the next cellular structure, the ribosome. So ribosomes are these pretty small little protein factories in our cells. They're made out of 2 components, the small and large subunit. We'll see this in more detail when we get into gene expression. And they're found either free within the cytosol of the cell or attached to this rough endoplasmic reticulum, which we'll talk about shortly. Here we see a transmission electron micrograph here, free ribosomes. They're very dark in this image here, so little black dots. And then here are these membranes we're seeing of the endoplasmic reticulum and some ribosomes bound to that. Ribosomes are mostly made out of that ribosomal RNA. There's proteins on them as well, but here we see the RNA folding up and coiling, forming that ribosome structure. All right, a quick and hopefully easy question. A cell with more ribosomes than an average cell might specialize in making what? Another component or set of components really in cells is called the endomembrane system. So here are these packages, compartments really within the cell that are membrane bound. This includes the nucleus that we just talked about. This helps move and package material around within the cell. So we'll talk about each one of these in turn. Yeah, even outside the cell or the outer surface of the cell, the plasma membrane is part of this system really, even though it's not inside endomembrane. All right, so material gets moved around through these little transport membrane bound structures called vesicles. And those will move material between these different structures. The endoplasmic reticulum accounts for a lot of that membranous stuff in eukaryotic cells. There's two kinds of ER, the smooth and rough. They're smooth and rough because one lacks ribosomes, that's the smooth, and one has ribosomes all over it. So by electron microscopy, that rough endoplasmic reticulum is going to look like this in this EM here, those little black dots with our ribosomes. Here's the smooth ER, no ribosomes. What does the smooth ER do? It's important for synthesizing lipids, carbohydrate metabolism, detoxification, and calcium ion storage. How about the rough ER? Well, it's covered with ribosomes. So if ribosomes are those protein building machines, the rough ER is really important in protein production. And specifically proteins that are going to end up on the surface of the cell or be secreted by the cell. So we'll see how that works in a second. This is also the membrane factory of the cell. One of our other membrane-bound organelles of the endomembrane system is the Golgi apparatus or Golgi apparatus. And I like to think of this as the shipping and receiving center. So as proteins or other compounds are made in other organelles, maybe specifically a lot of them in the rough endoplasmic reticulum, as well as the smooth, these will be processed in the Golgi apparatus. Maybe their proteins are cut or some kind of molecule is added to them. They're packaged then together in certain ways that will then direct them to where they need to go. So it's like the FedEx of the cell. It's the shipping and receiving. I encourage you to check out this animation here. I think this really shows this dynamic process of what the Golgi does. What's happening on this one face, it's called the cis face. This is the face that's closer to the ER or the nucleus. As these new vesicles come together, they're making new cisternae. So these guys will add together and they'll make one, and more will come together, they'll make a new cisterna. What's happening on the trans face, the shipping side, these cisterna are breaking apart and budding off and forming these other transport vesicles to send this material to wherever it needs to go. And so this cisterna is getting smaller and smaller and disappearing as new ones are being made on this other side. Lysosomes are a product of the Golgi apparatus and part of this endomembrane system. And these are really cool. These are like a little transport vesicle, but they contain digestive enzymes. So these are the little like digestive organelles of the cell. And we can have a look at what that might look like here. We see a food vacuole food being taken up by this cell, what will happen is this lysosome will fuse with the food vacuole. Now it's mixing in these digestive enzymes with that food, and it gets broken down. Now those little bitties can be maybe moved out of this vacuole or transported somewhere else. This slide shows that same thing that we just talked about, digestion here. But lysosomes are also important for breaking down old dead organelles. So here's an old mitochondrion here. It will fuse with that mitochondrion and break down that organelle. And those parts now can then be recycled. Let's look now at how a protein or group of proteins might be secreted from the cell, or maybe even end up in the cell membrane. So in the nucleus, a gene is switched on. Out comes a piece of messenger RNA. It gets translated by one of these ribosomes to build a protein at the rough ER. So we're building a protein now, and we see here's a little cluster of these purple dots where those are our proteins. Here they are moving to the Golgi, where they'll be processed, modified, repackaged, and out comes over here. This was our example of our lysosome, but here are this other proteins over here. These are intended to be secreted by the cell. So then from the Golgi apparatus, that vesicle, transport vesicle fuses, or secretory vesicle fuses with the plasma membrane, becoming one, and then releasing the internal contents to the outside part of the cell. If these were proteins that needed to end up in the cell membrane, they would already be embedded in this transport vesicle membrane. And when this fuses with the plasma membrane, then that just becomes the outside of the cell. So that's how this endomembrane trafficking works. All right, so given the choices in this question, what's the correct order in the secretion pathway? All right, a few more organelles. The vacuoles. So these are a wide range of different kind of compartments. And these are usually large vesicles. So vesicles are those little membrane-bound organelles, but these tend to be much larger. They're derived from the ER or Golgi, and they provide an environment within them that's different from what's in the cytosol. Just what we've already seen, a food vacuole, for example, is formed by phagocytosis when the cell eats. So it takes in a food particle like we see in this cartoon. Here's that lysosome interaction. We've already seen that. So that's an example of a vacuole. Other vacuoles, though, are like this, a contractile vacuole. Maybe you've seen this in sort of the classic paramecium model. This is important for regulating water content in cells. So here's a single-celled organism. It lives in the water. Water is going to rush into this organism because it lives in a freshwater environment, and water is moving down its gradient. And so this little single-celled organism doesn't want all that water. It has to expel it. So the water gets shunted into this contractile vacuole and then expelled back out. And so this is a way that these single-celled organisms can regulate the amount of water that is within. Another kind of vacuole, which is pretty big in plants, is this central vacuole. So this is found in many kinds of plant cells. This can be important for storage of different kinds of ions. If you think about the turgor pressure in a plant, so this filling up with water gives those plant cells that rigidity. And so those are some of the functions of this central vacuole. All right, so that's it for part one. We'll talk about mitochondria and chloroplasts and some other organelles in part two.